Hello everyone, today I am honored to introduce the research results on the changes in immune characteristics of acute, subacute, and chronic brucellosis.
Brucellosis is caused by Brucella, which affects livestock worldwide and is one of the most important zoonotic diseases in humans. Brucellosis patients present with a wide range of clinical manifestations, from asymptomatic to mild-moderate disease, and some patients may develop severe diseases involving multiple organs, even leading to death. A detailed investigation into the immune response of human brucellosis is still lacking. Single-cell transcriptome sequencing is a powerful technique for breaking down host immune responses, which has been used for various infectious diseases but has not yet been applied to brucellosis. We described high-resolution transcriptomic changes in peripheral blood immune cells at different disease stages and emphasized the relationship between disease stages and host immune response. After filtering the data, a total of 290,369 cell transcriptomes were retained. Each PBMC sample produces an average of approximately 8,799 cells. 9 major cell lines have been identified. 32 subtypes were identified, covering various immune cell clusters in peripheral blood. It is worth noting that most B-cell subsets are associated with patients with chronic brucellosis, especially plasma cells and plasmablasts. Association and enrichment of memory B cells in patients with chronic brucellosis. These findings reveal the link between B-cell clusters and disease staging. Based on the expression of cytokines and inflammatory genes, we assign cytokine scores and inflammation scores to each cell subpopulation separately, and used these two interrelated scores as indicators to measure the potential contribution of each cell to the inflammatory response in brucellosis. 6 subtypes of CD8 T cells were identified. It was found that Th1 cells have the highest exhaustion score in patients with brucellosis and highly express multiple inhibitory receptors, indicating the depletion of Th1 cells in brucellosis, especially for patients in the chronic phase. This may be related to inadequate control of persistent Brucella infection. MYT and gamma-delta T cells have low CD8 gene expression and are classified as CD8 T cells for transcriptome analysis. CD8 T cells further aggregated into 9 subgroups. Each CD8 T subtype exhibits different disease preferences. Immature clusters are enriched in healthy donors, while the rest are mainly found in acute and subacute patients. These data suggest that the decrease in cytotoxic T cell frequency in chronic patients may be related to the persistence of Brucella. The further aggregation of NK cells leads to the subdivision into 4 different subtypes. Multiple activation markers were found to be enriched in NK cells of Brucella patients, indicating that activated NK cell response is a significant feature of Brucella patients, especially in the acute phase. We investigated the phagocytic activity and antigen presentation ability of MDCs against Brucella infection. Only acute patients exhibit significantly higher phagocytic ability than healthy donors. In the presence of non-specialized antigen-presenting cells such as B cells and monocytes, we observed a significant decrease in phagocytosis, antigen presentation, and cell apoptosis ability in chronic patients compared to acute patients. The first use of single-cell transcriptome sequencing analysis revealed different immune responses in the acute, subacute, and chronic stages of Brucella infection. The characteristic of acute brucellosis infection is cytokine storm. The main characteristics of infection in the chronic phase of brucellosis are Th1 immune response imbalance and widespread depletion of T cells and NK cells.